Dear students, good morning. Hope you are very fine. Today's our offline class is about bullet groups. There are certain molecules on the surfaces of all cells in the body that can be recognized as foreign by the immune system of another individual and hence can induce the immune system of later. These molecules are known as antigens. As part of the immune response, particular lymphocytes secrete a class of proteins called antibodies that bond in a specific fashion with antigens. The membranes of red blood cells (RBCs) also possess several antigens. Depending on the nature of antigens present on the membrane of RBCs, various types of blood grouping has been done. Two such groupings, the ABO and RH, are widely used all over the world. Now, ABO grouping. Carl Landsteiner in the year 1901, along with his co-worker, recognized four types of blood groups in human beings, commonly known as ABO blood grouping. ABO grouping is based on the presence or absence of two surface antigens on the RBCs, namely A and B. Similarly, the plasma of different individuals also contain two natural antibodies called anti-A and anti-B. According to Landsteiner law, if an antigen is present on the RBCs, the corresponding antibody must be absent from the plasma. It also states that if the antigen is absent on the RBCs, the corresponding antibody must be present in the plasma. The immune system exhibits tolerance to its own red blood cell antigens. For example, persons with A group do not produce anti-A antibodies. However, they do make antibodies against the B antigen, and persons with B group make antibodies against the A antigen. Persons with AB group develop tolerance to both of these antigens and thus do not produce either anti-A or anti-B antibodies. People with O group, by contrast, do not develop tolerance to either antigens. They have both anti-A and anti-B antibodies in their plasma. Transfusion reactions. In many clinical conditions, blood transfusion is needed. In such cases, the donor's blood and a recipient's blood must be compatible to avoid severe problems of clumping, that is destruction of RBCs. A major cross match is made by mixing serum from the recipient with blood cells from the donor. If the blood groups do not match, for example, if the donor's blood group is A and the recipient's blood group is B, the recipient's antibodies attach to the donor's RBCs and cause clumping, that is agglutination. Transfusion errors that result in such agglutination, agglutination can lead to blockage of small blood vessels and cause hemolysis, that is rupture of RBCs. Therefore, the blood of a donor has to be carefully matched with the blood of the recipient before any blood transfusion. The below table shows blood group, antigens on RBCs, antibodies in plasma, donors group. Blood group A, it has A antigens on RBCs, antibody B in plasma, donors group is A and O. Blood group B, it has B antigens on RBCs and anti-A in plasma, donors group is B and O. Blood group AB, it has both A and B antigens on RBCs but no antibodies in plasma. Donors group is AB, A, B and O. Blood group O, it has nil antigens on RBCs but both A and B antibodies in plasma. Donors group, that is O. From the above mentioned table, it is evident that blood group O can be donated to persons with any other blood group, and hence O group individuals are called universal donors. Group O red blood cells lack A and B antigens, therefore the recipient's antibodies cannot cause agglutination of the donor's RBCs. Persons with AB group can accept blood from persons with AB as well as the other groups of blood. This is because blood group AB lacks anti-A and anti-B antibodies and thus cannot agglutinate donors' red blood cells. Therefore, such persons are called universal recipients. Thanks.